when we develop guideRNAs or choose which type of Cas protein we want to use to target a particular region in the genome, it really depends on the specific experiment that you want to do. Do you want to make very specific edits? Do you just want to make a cut and disrupt something? Do you want to insert a gene? Then specifically for this guideRNA, which gene do you want to target? Which part of that gene is important to you? Do you want to focus on a very specific part of a gene, or does it matter to you? So all these things need to be taken into account before you actually start designing a guideRNA or even choosing a Cas protein, because different Cas proteins can, for example, target different regions in a gene. Once you have determined your target, so which piece of the gene that you want to mutate, you have to choose your application. So, for example, if you have a Cas9 protein, you can modify this Cas9 protein to make a base editor. So instead of simply cutting the DNA in that particular case, you are modifying specific residues in the DNA without making a cut. So these are all types of choices that you need to make at that point. What type of edit do I want to make? So in terms of Cas nucleases, there's a huge variety of Cas nucleases that are being discovered, being described. Actually, almost every month there's a new publication on a new nuclease. However, only few are used regularly in the lab. The most popular one is Cas9, and this one is used routinely in the lab, is also modified to make, for example, base editors. There are some other Cas nucleases which are popular. For example, Cas12 proteins. In our experience in plants, they are less efficient compared to the Cas9 protein, but they can have other interesting features. For example, they can target other sites of the genome that Cas9 cannot target. If we want to target a gene, and if we want to make a choice for a Cas nuclease, each Cas nuclease recognizes the DNA with a specific RNA molecule that we call a guideRNA. So it's actually 20 nucleotides of RNA, that binds the DNA. When a Cas protein finds or has to look for a region in the DNA to cut, it actually uses an RNA molecule that is complementary to the DNA molecule. And actually, adjacent to that complementary RNA molecule, Cas proteins need some type of code, some type of sequence just next to it to be able to cut it. And this differs for different Cas proteins. For example, for Cas9, this is an NGG signature. For the Cas12, it's something else. It's more T-rich, so instead of G-rich. So this allows you to target different regions in the genome. If you want to target a specific region, you need to know the sequence context of that region, and this will allow you to select a Cas protein, because some Cas proteins, for example Cas9, they need a specific sequence, an NGG, so a G-rich sequence, next to the target site in the gene that they will recognize. Other Cas proteins, like Cas12, require a T-rich region next to the target site. So this will allow you to make a choice of which Cas protein to choose. Again, it depends really on your application. If you want to make simply a knockout to disrupt the gene, you basically take a region that is accessible for the best performing Cas nuclease, right? Because you just want to cut and get over it. The difficulty starts when you have a very specific region to target. You want to make a very specific edit, and you only have a window of, let's say, 4 base pairs. So then you really need to consider carefully, can I use, for example, Cas9? Do I need to go for a Cas12? Is it better to use a base editor? So all these things you have to take into consideration at that point. Sometimes you make a construct and it's simply not functional. So Cas proteins use a specific RNA molecule to recognize a DNA target. This piece of RNA that recognizes the DNA is only 20 nucleotides long. So you can imagine for even a single gene that you can design sometimes over 1,000 different targets. Not every target is as efficient as the next one. There is some variability. Some will not be able to recognize the DNA, well, not allow Cas9 to recognize the DNA and make the cut. Some will be highly efficient. There are some prediction programs that give a ranking and score this, but essentially you have to make the construct and see for yourself if it works or not. So this can be an issue. Also, some Cas proteins are not as efficient as another one. For example, Cas9 proteins, in plants at least, are more efficient compared to the Cas12 proteins. When we want to design a guide RNA, actually we have a lot of different online tools to do this. In these online tools, you basically click on the genome of the species that you want to study. In our case, it's often Arabidopsis. And you paste your gene sequence or the region in the genome that you want to target specifically also in this tool. You click submit and you get a list of, let's say, 100 targets that are possible within this region. And then you get some extra scores. For example, there are some bioinformatic tools that rank guide RNAs, and so you can make a more informed choice if this could be a good performing guide RNA. Something that is important as well is that the target that this tool gives you does not bind on other sites of the genome. And so this guide RNA that recognizes the genome is 20 nucleotides. So it is possible that there are some other sites in the genome which are very similar to the 20 nucleotides of your specific target, of your specific gene. And you, of course, usually only want to make that one single cut in that one single gene. Some of the guide RNA prediction programs that we often use are called CRISPR or FlashFry or CCTOP, but there are several others. And actually most of them work in a very similar way. So there's not really a single program that outperforms the others.
We usually design multiple guides per gene, and we make combinations of those guides. So for example, if you want to screen 6 genes, we will design 3 guides per gene and then combine these together and see which ones work best. Other issues are often the cloning. So what's different with single targeting is you have to have multiple guides in one vector. So then you have to clone 6 different guides in the vector, and sometimes that is an issue. Sometimes a problem that we encounter is cloning the vectors. So instead of cloning 1 guide in your vectors, you have to clone 6 or 60. Sometimes there can be issues with the cloning. I recently tried to make 60 guides for 1 vector, but there I failed to make the vector. Yeah, and maybe the efficiency is too low, or no. It just didn't want to come together. 12 worked, so 12 guide RNAs for 1 vector worked fine. 60 didn't.
The efficiency of the guides, the thing is, it's kind of difficult to predict what the efficiency will be. So you take your gene and you put it through the software, you get some output, but it doesn't necessarily always reflect what you will get as the final result. Because one of the difficulties with these programs is that they're based on mammalian cell line data, and they're not readily transferable to plants, actually. Right, it's very difficult to predict, actually. So at the moment, I'm working with an array of 6 different guide RNAs cloned in one array, and for one of the guides we have 0%, for the other one 75%. So it really depends a lot on the guide that you actually use. One of the difficulties with maize specifically is the transformation protocol. So before I worked with rice, and there you can start from mature embryos, mature seeds. But in rice, typically they use immature embryos, and it's quite labor intensive. You also need a lot of greenhouse space to actually get enough embryos to start your transformation protocols with.